Good morning, students. Hope you are all fine. So, in our previous video, we have talked about the Illumina sequencing. We thought that yes, we will be discussing more on the Illumina sequencing in our next video. And now you have got some idea, basic idea that how Sanger sequencing can be done, how the pocket sequencer, like the Oxford nanopore, can be uh, planned to execute the sequencing for specific purpose. Now in this video, I'm going to discuss in details along with one important video from the website that how the Illumina sequencing can be done. Now, you know what? Illumina sequencing is a very uh, important platform for the sequencing of nucleic acids. And the Illumina sequencing is actually known that is sequence by synthesis method, which is one of the important and widely used next generation sequencing NGS platform that allows very high throughput, I mean high quality, high fidelity, high throughput DNA sequencing mm, with high accuracy and also with the high scalability. So to do the Illumina sequencing, there are certain important steps that we need to follow. And let me discuss first, and then we will actually run one Illumina sequencing video that have been downloaded from the Illumina platform itself. So first thing that we have to keep it in our mind that the Illumina sequencing has to do first with starts with the library preparation. First, we have to do a library preparation. So first, the DNA that we want to sequence, the DNA has to be extracted and then fragmented into some smaller pieces. Now, suppose you have a very large size of DNA, and then you have to cut the DNA into several fragments of small pieces. Now, each and every small pieces of the DNA has to be added with the adapter with some unique barcodes. So these small adapters with the unique barcodes are then ligated to each and every ends of the DNA fragment. Suppose you have a DNA fragment, it has got two ends. So each of these two ends, the adapter has to be ligated. These adapters have the sequences that are required for binding to the Illumina flow cell for the sequencing to proceed. So in the Illumina, that is the Illumina platform, we call it a flow cell, and flow cell has got certain sequences. So the small DNA pieces uh, that has to be ligated with the adapter in each of these two. So these two adapters have the complementary sequences of the small sequences found on the entire flow cell of Illumina platform. Then second, what we need to do, we have to do a cluster preparation or cluster generation. So the prepared DNA fragments are then immobilized or attached on the solid support called the flow cell. We call it a flow cell through attachment with the adapters. So it's just about what? It's also about suppose you want to join two pages, or you want to fix your photograph on a page. So you are actually adding some glue. You are adding some glue at the back of the photo. You are adding some glue on the pages where you need to fix, and then you are pasting. Same here. So in the piece of the DNA, you have two different adapters on the opposite side, and the complementary sequences are actually attached throughout in the flow cell. So these DNA are ligated with that, and with the complementary nucleotide binding, all the adapters along with the small pieces of the DNA will actually bind with the flow cell. So the DNA then clusters are actually formed with the bridge amplification strategy that you will see in the video that will play you for. And during each of the DNA fragment is then cloned and then amplified, creating lots and lots and lots of clusters of this identical sequences. Now, as I mentioned that this sequencing, Illumina sequencing is known as the sequence by synthesis method. So suppose you have the flow cell in the present on the plot. Suppose you have the flow cell like this. Your flow cell has specific adapter. So your flow cell has got specific adapter. Your DNA will bind here with the adapter. So your flow cell have an adapter, and your DNA will actually bind. Your flow cell have got some small sequence. Your small pieces of the DNA along with the adapter will bind with that, and then by taking this, we are actually adding the fluorescent probe tagged the nucleotide that is A, T, G, and C, and then these all will actually have a sequence. So we call it a sequence by synthesis method. So the flow cell is now loaded onto the Illumina sequencing instrument. After the flow cell, it will actually end into that. And finally, the fluorescently labeled nucleotide that is A, T, G, and C are added into the flow cell, and their incorporation is detected by when there is a real-time sequencing will proceed. So each nucleotide is tagged with a different fluorophore. Now there are 4 different nucleotides A, T, G, C. These 4 different nucleotides are tagged with 4 different types of fluorophore, allowing them proper incorporation as well as the identification. So where the A is being added, where G is being added, as there are 4 different nucleotides, so it is very easy to then identify that, because each and every nucleotides have a specific absorption, and then on the basis of the chromatogram, we can find out that if A is added, there is a specific signal of fluorescence. If T is added, there is another amplification or another uh, uh, mode of this absorption of the fluorescence. I mean, basically a specific fluorescence. So A, T, G, and C have different fluorescence signal. Then detection of the imaging, basically a large uh, uh, excites of now when the fluorescently attached this ATGC will actually bind. So they actually have a specific signal. A laser is there. The laser will actually excite the fluorophore on the cluster, and there is a small camera, high throughput camera, that actually captures the images of the fluorescence signal. So যে fluorescently tagged ATGC add হচ্ছে তার রেসপেক্টে যে সিগন্যালটা পাওয়া যায় সেই signal has been captured by a camera. So the intensity and the location of the emitted light are used to determine and identify that which nucleotide has been incorporated at which position of the sequence. Now, when different nucleotides have been added and we have captured some sequences through the camera. So finally is the base calling. So base calling is that is statistical significance that yes, if a signal is found, how is the statistically significant that yes, this is A. So that's why base calling is an important step. So the raw images are processed through a base calling software, which convert the fluorescent signal into a nucleotide sequence data. So it's basically the conversion of the signal into the sequencing data. So the step assigned the correct base out of A, T, G, and C to each cluster of the flow cell. And finally, when we actually gather lot of data, then there is a data analysis. So the generated sequence data is now processed through the in silico biologist or bioinformatician uh, uh, using some bioinformatics tool to obtain the final sequence data in FASTQ format. The step assigned the correct. So the sequence reads are often aligned. Now, once we have the different short sequence read, then you need to align the sequence. Then you need to align the sequence. Suppose there is a piece of page where many things have been written. So you actually tear the pages in 4 different parts like this. Now, if somebody will tell you that now you have just tried to align this, so you will find that after which sentence, so this piece and this piece can be aligned properly. So final step is basically to align the sequence for further analysis. And finally, the high quality control steps will be performed to assess the accuracy and reliability of the data produced. So now after that, if you I I would like to play this video for you so that you can actually have some video tutorial. Whatever I said, you can correlate with that. So please uh, uh, see this video here. I'm playing for you.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced
As I mentioned, you can see that a piece of the DNA are ligated with two different adapters because a piece of the DNA has two ends. So two different adapters are being ligated at every end of the DNA fragments.
cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
So this is basically the flow cell, and the flow cell consists of arrays of different oligonucleotide sequences. Arrays of oligonucleotide having the complementary sequence of the adapter that we have already ligated with the pieces of the DNA that we want to sequence.
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with alon, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in
Will
clonal
actually produce
amplification of
lots of
all the
clusters
fragments.
with the bridge amplification method.
After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3'
All
ends
the
are
reverse
blocked
strands
to
are
prevent
washed out from the
unwanted
cell
priming.
by washing, and then the forward strands kept there. Now this will
Sequencing
actually
begins with the
act
extension of
as a
the first
template
sequencing
for
primer to
further
produce the
extension.
first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected.
So basically this
The
process
template now
continues
folds
over and
over and
over and
binds the
over.
second oligo on the flow cell. Index
One
2
and
is read in the same
each and
manner as
every
Index
nucleotide is
1.
being added. The
Polymerases
polymerase will
extend
actually
the second
extend.
flow cell
So it's a
oligo, forming a
sequencing.
double-stranded
We are
bridge.
doing the
This
sequencing
double-stranded
by
DNA is
synthesis
then
method. The strands
linearized
are being
and the 3'
synthesized,
ends are
and the
blocked.
addition of
The
each and
original
every
forward
nucleotide with
strand is
4
cleaved
different
off and
fluorescent
washed
nuclear
away,
probes.
leaving only
The
the reverse
signal is
strand.
captured by the
Read 2 begins with the
camera.
introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The read-to
For
product
that,
is then
we are
washed
actually
away.
gathering lots and lots of sequence.
This entire process generates millions of reads, representing all the fragments.
Absolutely.
Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The
So it's
paired
the
end
part now.
information
Now this is the part for the bioinformatician who will actually do the data analysis part of the sequences. So with the different programming like R programming, Python, and others. So this alignment that can be done. There are a lot of other platforms also that can be utilized for aligning proper alignment of the sequence reads to a specific reference genome from the database found.
is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
So this is how actually the NGS, the next gen sequencing, that can be platform. And you know what? Nowadays, I mean, the present generation is basically the generation of the in silico biology, where both wet lab work and the dry lab work and sequencing is a major part of different research laboratories. So hope you have uh, 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 found interest in this sequencing. And next in my class, I will actually try to discuss different other sequencing platform like pyrosequencing, Sorex, and others in my second next class tutorial. And by the time, so it's so have a nice day and bye.